If you remember, in the first video, we have analyzed cell as seen with a light microscope. And today, we're going to see the structures you can find inside the cell when you use electron microscope, which has greater magnification power than the light microscope. It's very important for you to bear in mind that cell is a very tiny structure which you cannot see unless you use microscope. It's 5 to 10 micrometer structure, so therefore it's microscopic. You cannot see it with your naked eye unless you use microscope. So when you use electron microscope, you can see that the cell membrane we have described in the first video as a single layer. It's two-layer structure. It's like this. It's a two-layer. It's called phospholipid bilayer. It's a phospholipid bilayer, as I said, and therefore it contains two layers. This is an important greater detail you can see with the electron microscope, which you cannot see with the light microscope. The dark staining area we have at the center, which we call nucleus, has some pores attached to its membrane. It looks like this. On these pores, when you look at it, you will see that there are some pores. And the nuclear membrane, you will find out that there is a membrane surrounding the nucleus, and we call it the nuclear membrane. It's called the nuclear membrane. We call it nuclear membrane. If you can remember, in the previous video we have said that there are some debris inside the cytoplasm, which we call the cytoplasmic granules. But when you use electron microscope, you can see that those granules are certain structures that can perform various functions inside the cell. And one of these structures is an endoplasmic reticulum attached to the cell membrane. We call it endoplasmic reticulum, and it is attached to the cell membrane like this. It's continuous with the cell membrane. Excuse me. And sometimes you will see some granules attached to the endoplasmic reticulum, giving it characteristic rough appearance. And we call these granules ribosomes. They are attached to the endoplasmic reticulum, giving it characteristic rough appearance. And it's called ribosome. And for this reason, we call it rough endoplasmic reticulum, because it contains, because there are ribosomes attached to it. We call it rough endoplasmic reticulum. Rough endoplasmic reticulum. It's called rough endoplasmic reticulum. Then at the center of the nucleus, there is usually a dark staining area. Which is called the nucleolus. We call it nucleolus. It's called nucleolus. And between the nucleolus and the nuclear membrane, there is a space, just like a space between the cell membrane and the nuclear membrane. And we call this space nucleoplasm. Nucleoplasm. It's called nucleoplasm. We call it nucleoplasm. As I said earlier, there are pores surrounding the nuclear membrane entirely, and these pores are called nuclear pores. We call them nuclear pores. This is the cell membrane, which seems to be a membrane containing two layers. There is another endoplasmic reticulum which do not possess ribosome. That is to say, there are no ribosomes attached to it, and we call it smooth endoplasmic reticulum. We call it smooth endoplasmic reticulum. They are called smooth endoplasmic reticulum. Smooth endoplasmic reticulum. There are no ribosomes attached to it. Then somewhere in the cell you will see some structures looking like this. And we call them mitochondria. Cell contains numerous mitochondria depending on its energy requirement, because this produces energy. So therefore, they are numerous in number. We call them mitochondria. They are called mitochondria. There is another important structure which is called microtubule. We call it microtubule. These microtubules actually they play cytoskeletal role. That is, they give support to the cell. We call them microtubules. We also have lysosome. Another important structure you can see is the Golgi apparatus or Golgi complex. If you like, you can call it Golgi body, Golgi complex or Golgi apparatus. We call it Golgi apparatus. It's named after the Italian scientist called Camillo Golgi, who was responsible for its discoveries. It's called Golgi apparatus. An Italian scientist in person of Camillo Golgi was the person responsible for its discovery. These are some of the most important structures you can see using electron microscope. And we are going to explain the function of each and every structure you can see here in the next video. Endeavor to watch the next video.